So we've talked about the overall photosynthetic process. We've talked about the nature of light, and we've figured out how we're able to convert light into usable cellular energy, but we've yet to build the sugar. So this lecture is on how we're going to take the individual steps of the Calvin-Benson cycle, the products and reactants, and figure out how this whole thing's working. The Calvin-Benson cycle is otherwise known as the dark reactions of photosynthesis, or the light-independent reactions of photosynthesis, or just the Calvin cycle. It could be any of those. I mean, there's lots of things this is known as. This Calvin-Benson cycle is going to take atmospheric carbon dioxide, so the CO2 that's in the air that you have produced, and you're going to attach that CO2 up to other molecules to produce large macromolecules, specifically glucose. That's called carbon fixation. You're taking carbon dioxide out of the air and fixing it. By fixing, I mean sort of like affixing or gluing. You're taking CO2 out of the air and gluing it to something else. That's the point of this part. Pulling in that CO2, gluing it to another molecule. Building these organic molecules requires a huge amount of energy, an enormous amount of energy. These reactions are light independent. That means they do not have to happen in the light. So where does the energy come from? The light-dependent reactions. In the light-dependent reactions, you produced a lot of two products. What were the two products? ATP and NADPH. Both of those have superexcited electrons. And we can take those excited electrons and make them do work. For every 6 carbon dioxide that enter the cycle, you're going to use up or hydrolyze 18 ATP and 12 NADPH. It's a lot of energy. What does it mean to hydrolyze something? You're cutting it using water. It's hydrolysis. And what happens when you cut something? You release energy. And now you're going to use that energy to make glucose. There are 3 steps. 3 phases: carbon fixation, reduction, or otherwise known as carbohydrate production, and the regeneration of ribulose bisphosphate, or RUBP. So 3 phases: carbon fixation, reduction, regeneration. I'm going to go through each of those phases in detail. But please note, it is a cycle. You're going to start with the same thing you end with. So fixation, reduction, regeneration. What do I mean by reduction? Gaining an electron. So step 1, carbon fixation. In carbon fixation, you're going to take 6 molecules of carbon dioxide and 6 molecules of ribulose bisphosphate, otherwise known as RUBP. And they're going to build 12 molecules of what's called 3-PG. Now, you don't need to know all of that. The terms you need to know are RUBP, carbon dioxide, and take a look at this molecule. It's got 3 carbons to it. That's going to come into play later. It's just sort of like foreshadowing. So you've made, you're taking these 6 CO2, 6 RUBP, and building 12 molecules of this other of 3-PG. The deal is, whenever you take 2 molecules and put them together, is that an anabolic or a catabolic reaction? It's anabolic. Endergonic or exergonic? Endergonic. So you need energy. You need energy for this reaction to happen. You need a lot of energy. How would you reduce the amount of energy you need? You add an enzyme. You add a catalyst. In this case, the catalyst or the enzyme is a molecule by the name of ribisco. Ribisco is actually ribulose bisphosphate, so RUBP, carboxylase oxygenase, which means it adds carbon dioxide to ribulose bisphosphate. It grabs carbon dioxide, takes RUBP, and squishes them together. That's its job. It is very, very likely the most abundant protein on the planet. Every plant has it. Every photoautotroph has it. So Rubisco is an enzyme, and it behaves just like any other enzyme would. So we've step one, carbon fixation. We've taken carbon dioxide out of the air. We've attached it to RUBP. We've made 3PG, and that can go on. The products from that step are going to go on to be recombined to form, effectively to go on to form glucose. You're going to take these 12 extra molecules, break them apart a little, and recombine them. And now you've built one molecule of glucose. The big thing to note is that RUBP you started with is now broken. But you do have sugar. So we've gone from taking carbon dioxide out of the air to combining it with RUBP, breaking that apart to build glucose, and then now we've gotten our glucose. But we have a bunch of broken down RUBP. So that's step two, carbohydrate production. The third step is now that you've broken all that RUBP, you need to build it back again. Because it's a cycle. You have to end with what you started with. And what we started with was RUBP. So we're going to apply a bunch of ATP to produce that RUBP again. We're using the ATP we created in the light-dependent reactions to rebuild our RUBP. So we have three steps. Fixation, carbon dioxide out of the air, attach it to RUBP. Step two, take that product, break it down to form glucose. But you've broken the RUBP. So step three is rebuild your RUBP, and that requires energy. Yes? Can I draw it? In fact, let me just switch the slide because I'm pretty sure it's on the next slide. There you go. You don't like that drawing? Carbon fixation. You're taking carbon dioxide out of the atmosphere and you're attaching it to RUBP. Step two, you're taking that combined product and you're breaking it, and that produces glucose. But you have now broken the RUBP. So step three is rebuild the RUBP so you can go back into the first phase. It's a cycle that continues. So the net results are you've taken carbon dioxide out of the air, you've reduced it and formed glucose, and you've recovered your RUBP. This is going to, that recovery of the RUBP is going to require a lot of extra energy from the light reactions. We've gotten through the light reactions and the dark reactions now. We focused on that Calvin-Benson cycle, the dark reactions. These happen if light's there or not, and it is a three-step process. Fixation, where you take carbon dioxide out of the atmosphere and attach it to RUBP. The molecule that does that is Rubisco. It's an enzyme that can grab CO2 and attach to RUBP, two distinct things. Reduction, or carbohydrate production. That's where the glucose is going to be built by breaking apart the RUBP CO2 complex, using those parts to build sugar. And then you need your third step, which is the recovery of or regeneration of RUBP, ribulose bisphosphate, in order to get this process to start again. And it's that last step that's going to require most of the energy from the light reactions. Remember, the dark reactions are powered by the ATP produced during the light reactions. There we go. Light reactions plus dark reactions, we end up producing oxygen and sugar. These content review questions should help focus your studies. Moving into the next set of lectures, or the next mini lecture, we're going to talk about
what happens in this bizarro world where it might be really hot and Rubisco doesn't work terribly well. So we're going to talk about different types of photosynthesis, um, specifically differences between C3, C4, and CAM photosynthetic models. It's kind of interesting. Stick around. We'll learn about how cactuses work. You'll see some pineapples growing, and it's weird because people see pineapples growing and they don't grow in trees; they grow on the ground. So it's exciting and new. Or maybe you already knew that, but stick around anyway.